Researchers made a mouse embryo grow 6 legs instead of the usual 4, and some internal organs ended up outside the body. Today we are bringing an interesting video which will truly amaze you. Before we start, if you still haven't subscribed to our channel, make sure to hit the subscribe button and turn on the notification bell so you never miss out on our latest videos. In today's episode, we're diving into an intriguing application of 3D technology in scientific research. Specifically, we'll explore how 3D reconstruction plays a pivotal role in understanding complex biological processes. Introduction to 3D technology. In the vast landscape of technological advancement, the journey of 3D technology has been nothing short of revolutionary. It dates back to the early 19th century when stereoscopic photography was invented, allowing for images with depth perception. Fast forward to the mid-20th century and the emergence of computer graphics paved the way for the digital era of 3D modeling. But have you ever wondered how these advancements have impacted fields beyond entertainment? Let's take a closer look. 3D technology gained popularity in the late 20th century thanks to powerful computers and advanced software that enabled the creation of complex simulations and virtual environments. Today, 3D technology has permeated various fields, from entertainment and architecture to medicine and scientific research. In the realm of modern genetics, 3D technology, particularly 3D reconstruction and structure from motion (SFM), has become an indispensable tool in unraveling the complexities of biological processes. 3D reconstruction and structure from motion. 3D reconstruction involves the creation of three-dimensional models from a set of two-dimensional images or data points. Using advanced algorithms, this technology seamlessly merges the images or data points, resulting in a meticulous 3D depiction of the object or scene under examination. Structure from motion, SFM, is a powerful technique to reconstruct 3D structures from a sequence of 2D images. By utilizing the relationships between camera positions and orientations, SFM enables us to accurately estimate the geometry of the scene. But how do these techniques help scientists unravel the mysteries of biology? Let's find out. These technologies analyze visual information captured from multiple viewpoints and use computational methods to infer the spatial relationships between different elements. For example, in genetic research, confocal microscopy captures a series of 2D images of cellular structures at different focal planes. By applying 3D reconstruction algorithms, these images are combined to generate a comprehensive 3D model of the cellular architecture. This enables scientists to explore the intricate spatial organization of genes within the nucleus. Beyond genetics, 3D reconstruction and SFM have found applications in a multitude of fields. Archaeologists utilize these techniques to reconstruct ancient artifacts and archaeological sites, providing insights into past civilizations. In environmental science, researchers use drone imagery and SFM to monitor changes in landscapes, track deforestation, and assess biodiversity. These technologies are now essential for everyday applications like virtual reality gaming, augmented reality navigation, and 3D mapping for urban planning and architecture. Combining genetics experiments with advanced 3D technology shows how interdisciplinary collaboration can push the limits of scientific exploration. By using 3D reconstruction and SFM, scientists can explore biological systems in more detail, leading to important discoveries. Case study: the 6-legged mouse embryo. Our focus today centers on a remarkable study conducted by scientists at the Gulbenkian Science Institute in Portugal. Here, research conducted by developmental biologists Anastasia Lozowska, Moises Malho, and their colleagues made an unexpected discovery while studying the developmental stages of mouse embryos. By changing the TGFBR1 gene, which usually controls body development from the trunk to the tail, researchers made a mouse embryo grow 6 legs instead of the usual 4, and some internal organs ended up outside the body. Now, let's delve into this intriguing genetic anomaly. The TGFBR1 gene, also known as transforming growth factor beta receptor 1, plays a crucial role in embryonic development and various cellular processes. Understanding the genetic anomaly. Genetic anomalies showcased in the case study provided an invaluable insight into the role of DNA's 3D structure in development. The TGFBR1 gene is part of a gene family that creates receptors for the transforming growth factor beta, TGFB, signaling pathway. This pathway guides embryo cells to develop hind limbs or external genitals and controls cell growth, proliferation, differentiation, and programs cell death. During embryonic development, the TGF-beta signaling pathway plays a fundamental role in shaping various tissues and organs. TGF-br1 receives signals from TGF-beta ligands and uses them to control gene expression and cell behavior. When functioning correctly, the TGF-br1 gene helps orchestrate proper embryonic development by participating in processes such as cell differentiation, tissue morphogenesis, immune regulation, and extracellular matrix remodeling. In the absence of a functioning TGF-br1 gene, the embryos unexpectedly produced extra limbs, suggesting a deep interconnection between genetic expression and physical formation. Let's explore how 3D models shed light on this fascinating anomaly. To further their research, the team utilized advanced 3D computer vision techniques. They created a detailed 3D model of a mouse embryo with the TGF-br1 receptor protein deactivated. This model highlighted the developmental differences, showing normal limbs in turquoise and the additional limbs in magenta. These visualizations play a crucial role, not only in academic research but also in wider applications such as virtual reality, augmented reality, and computer graphics, elevating realism and improving the user experience across diverse fields. The team studied mouse embryos that were 10 to 17 days old, some with a working copy of the TGF-br1 gene and others without. The implications of this research extend beyond mere academic curiosity. Dr. Moises Malo and his colleagues believe that these findings could give us a better understanding of human development and help us explain why some animals, such as snakes, do not have certain limbs that most lizards have. The research also opens discussions on developmental plasticity, which could pave the way for addressing developmental disorders in the future. But as we embark on this journey of discovery, we must also navigate ethical considerations. Advancements in technologies like CRISPR-Cas9 provide new opportunities to understand and fix genetic anomalies. However, there are ethical concerns about the consequences of changing the basic components of life. Ethical considerations surrounding genetic manipulation are complex and multifaceted. Key concerns include the potential for unintended consequences, such as off-target mutations and ecological disruptions, stemming from techniques like CRISPR-Cas9. Enhancing humans raises questions about fairness, equality, and informed consent. Considering the historical context of eugenics, it's essential to avoid discrimination and promote diversity. Furthermore, genetically modified organisms have a significant environmental impact, and the long-term effects of genetic interventions on individuals and ecosystems present considerable challenges. Experts warn about the possible negative effects of genetic manipulation and stress the importance of strong ethical guidelines to oversee research and maintain responsible conduct. Finding a balance between scientific progress and ethics is important in genetic research. This ensures that we not only protect against potential misuse, but also promote equal and fair access to the numerous benefits that arise from such groundbreaking advancements. Technological impact and future prospects. The application of 3D technology in medical science, especially through the use of 3D computer vision, provides a richly detailed and immersive way to comprehend intricate biological structures and processes. This can significantly help to identify spatial relationships and the dimensions of structures, which are crucial in both educational and practical medical applications. Studying genes like TGF-br1 in embryonic development has great potential for future scientific research and medical treatments. Understanding the intricate mechanisms underlying normal development opens avenues for innovative approaches in gene therapy and developmental biology. Researchers can identify genetic factors associated with developmental disorders or congenital anomalies. This knowledge can help them create targeted interventions to treat or reduce these conditions. Moreover, studying genes like TGF-br1 can provide valuable knowledge for developing new treatments for many diseases, such as cancer, autoimmune disorders, and neurodegenerative conditions. So, what does this mean for the future of medicine? Let's find out. Advances in gene editing technologies have the potential to revolutionize the manipulation of genes linked to different disorders, providing personalized treatment options designed to match an individual's genetic profile. genetic research, developmental biology, and gene therapy together promise to bring about big advancements in medicine, giving hope for better health and quality of life for many people around the world. 3D technology is not just for video games or movies, as we saw with the six-legged mouse embryo. It is an important tool in scientific research, helping us understand the complex interactions in biology. Advancements in 3D visualization help researchers better address developmental challenges and diseases, making technology an essential tool in science. Challenges and limitations. While 3D reconstruction technology has made remarkable strides, challenges persist in its application to genetic research. One significant hurdle lies in accurately capturing and reconstructing complex biological structures with high fidelity. Obtaining precise 3D reconstructions of biological specimens, like cells and tissues, is challenging due to their inherent variability, irregular shapes, and dynamic behaviors. Imaging techniques like confocal microscopy can have limitations, such as low signal-to-noise ratio or photobleaching, which make the reconstruction process more complicated. To overcome these challenges, scientists are continuously refining imaging technologies and computational algorithms. Improved microscopy techniques like super-resolution microscopy and light sheet microscopy provide better imaging of biological samples, which improves the input data for 3D reconstruction. New computational methods are being created to automate and improve their reconstruction process. These methods, such as machine learning algorithms and deep learning approaches, aim to minimize the need for manual intervention while enhancing accuracy. Another significant challenge in genetic research is the interpretation and analysis of complex 3D datasets. Extracting meaningful insights from volumetric data requires sophisticated computational tools and expertise in bioinformatics and data visualization. Scientists are addressing this challenge by developing user-friendly software platforms and computational pipelines tailored to the needs of genetic researchers. That sounds absolutely captivating, right? If you are fascinated by the way technology is influencing our perception of the world, please feel free to share your thoughts in the comments section. It's always great to hear others' perspectives. Thank you for joining us on this journey through the world of genetics and embryonic development. If you have learned something new, remember to like, subscribe, and click the bell icon for more content on how modern technology affects our lives in surprising ways. Stay tuned for more cutting-edge research in the field of genetics and the technical world. See you next time.